When we think about gene therapy, I think the cleanest way to categorize gene therapies is through in vivo and ex vivo. In vivo gene therapy is sort of the most intuitive approach. It's what probably comes to your mind with gene therapy, where we are going to package a therapeutic gene into a viral vector and then inject the viral vector into the patient's body. Now, like I said, delivery is one of the big challenges. Ever since biologists started playing around with viruses in a lab, it became apparent to them, and they started publishing about this, that they could manipulate the viral genome and use the virus to dock up on cells and to cross the membrane and then cross the nuclear membrane and deliver a therapeutic gene either into the chromosomes directly or on a little extra chromosome region that we call an episome. But either way, the cell would then start producing the viral proteins in the cells. And in this case, it would be the therapeutic protein. So this has been the route taken by Novartis. They bought out Avexis, and they have an in vivo delivery system with Zolgensma for spinal muscular atrophy. And they inject directly into the patient's blood, little kids. And that is sufficient. The AAV, they use AV9, it will dock up on the motor neurons in the spine and punch the therapeutic protein into the motor neurons, and then they start producing SMN protein, and the motor neurons remain healthy. They don't die off any further. And so the kids maintain, muscular control to some degree. It's not perfect, but it does work. And then we saw, Spark do this with in the eye. They got Luxturna approved. That was an ocular injection. So this is another key piece, which I'll hit on again, is the route of administration. You can either go systemic into the blood, or you can go with a localized injection if that's the best way to hit your cellular targets. The eye is nice because it's separate from the immune system, and I'll talk about why that's a value. So that's a real quick nutshell on in vivo delivery. And there's a lot of steps involved with manufacturing AAV virus that is ready to go at the right dose to be injected into the patient. That's been a big challenge, and there's some talks this morning about how to help out the manufacturing system to make sure we have enough viral vector to satisfy the industry and the patients. So that's in vivo. Now there's this other category, which is ex vivo, and this is outside of life. This is outside of the human body, and this is proven to be incredibly effective. It's really a form of cell therapy. It's a form of cell therapy that involves genetic manipulation. That's the way to think about ex vivo cell therapy, and it's the way to think about CAR T as a treatment for cancer patients. CAR T is arguably the most successful version of ex vivo gene therapy. So what does this look like? You remove a cell population from the body. Okay. Now, this is most readily done with blood cells and immune cells. Anything that's circulating in liquid is going to be a good candidate to take out of the body and then keep surviving in a lab environment for a few weeks while you genetically manipulate them. And then you're in a position where you can reinfuse them back into the patient's body. And they're now like super. This step is most commonly done with lentivirus. It's another type of virus, and I'm going to explain why that's become kind of the industry standard. It's more optimal for these ex vivo approaches. This is done in a lab environment, so you get some benefits here in terms of watching the process happen. You can do a little bit of observation and sort of quality control before the cells are then infused back into the patient's body. So like I said, I work in the CAR T industry. This is done in about 3 weeks. And Kite Pharma, owned now by Gilead, they do this really well, I will say that. In terms of the speed with which they manufacture